Well, thanks very much. It's a great pleasure to be here, and particularly because I only live just at the top of Taylor Way, so it's not very far to come for this kind of an evening anyways, so that's great. First off, I do need to start with a disclosure to let you know that I have done some work with the pharmaceutical industry in the past, and where my grants come from, generally mostly from Ottawa, the Canadian Institutes of Health Research, and I do receive honoraria sometimes when I go to a conference. They help pay my way and those kind of things. Because conflicts of interest can be a problem in medicine, what I do to mitigate these conflicts is I limit my work with industry to medicines largely before they're released, so I'm not involved with marketing or advertising for medicines. I don't use any industry information in my slides. I make them myself. And I always tell people about my conflicts of interest before I start. So the things we want to talk about tonight, though, we'll touch a little bit on some medicines at first, because I want to talk about some older and some now, what I hope are new ideas about the brain, about the chemistry in the brain, how that relates to schizophrenia. That'll be sort of the kind of the science part of what we're going to talk about. Then we're going to talk more about clinical things and things in patients and so on, and about this idea that parts of the brain kind of shrink a bit in schizophrenia, and we'll explore whether or not that is sort of irreversible, like in some illnesses like Alzheimer's disease or so on, or whether schizophrenia is different in the way that we can do some things to change that around, which I'm really quite excited about, and we'll see if we can get interested too. So the first part is the kind of older and newer ideas about the chemistry of schizophrenia. Now, those of you who have family members, or if yourself you have schizophrenia, probably know about this chemical that's involved in the communication between cells. This is how our nerve cells communicate. This is one cell here. This is at a point of contact, that the cells have these points of contact between each other where information goes from one cell to the next cell. And the way that happens, the information is communicated by these things we call neurotransmitters. They're the messengers for the cells. And the particular one that's well known in schizophrenia is called dopamine. And I'll just do this little cartoon now. It's going to show you how the information cycles, where the medicines that our patients take work out here. But you'll see in this, I hope. That's how a message goes across, and then the messengers are brought back into the cell and packaged again for another episode of communication. And many of my cells are communicating right now in terms of that's how information goes between my cells, and I can convey it to you. You're listening. The same thing is happening in everybody's brain as you listen and so on. These messages go back and forth. And what we think is that in schizophrenia, actually, this is the conventional wisdom, this amount of message from the dopamine is like too much, okay? So too much is getting out. We'll go back, run that again.
It's going to shut off in just a second here. Yeah, sorry about that.
Let's go back these steps. So imagine this is the message coming out, and it's too much message going to that receptor, and then the message, the transmitters get pulled back in again and packaged. So what we do with our medicines, the antipsychotic medicines, they kind of block some of this communication because there's too much there in schizophrenia. Another reason, though, that we understand why the dopamine is likely to be a culprit is what some other drugs, street drugs, do to this dopamine system. So if we have amphetamine or cocaine or methamphetamine, what they do is make the messenger very high inside. Okay, so you saw that they push it out of the package, but the transporter that's supposed to take the messenger back in, they make that run backwards. So we get really a flood of this dopamine in the connection between the cells. And that can, not all people who take methamphetamine get psychotic actually, but some do. And we think that is because of this too much dopamine there. So knowing about two kinds of medicines, the one we use for schizophrenia to help it, that blocks this receptor, And by understanding how amphetamines, the stimulant drugs, work, it's given us, this is back 1960s really, this kind of information about how important dopamine is in schizophrenia. But we've kind of been locked in that really since the 1960s. We haven't had truly novel medications. We haven't had a lot of, they come up with different side effects, and that's helpful for patients, and we can get a right match of a medication and side effect, or the lack of side effects. But we haven't learned a whole lot more, really, to be honest about it. And this is why Tom Insel, the head of the National Institute of Mental Health in the U.S., said we've really got to rethink what's going on in the brain in schizophrenia to understand things a bit more. And I think we can go back, actually, even further to 1890. This is William James, who was a professor of psychology at Harvard. Came up with a lot of really important wisdom from that time, still stands up today. He kind of coined this term of plasticity. And plasticity, you don't necessarily need to think about a hard plastic like this laser pointer is made of, but think of Play-Doh. Think of it in terms of Play-Doh, it has a structure. That structure is weak enough so it will yield to an influence. You can mold it, but it doesn't just happen all at once. It doesn't suddenly change. It yields over time. And he was thinking about how habits form and these kind of psychological processes in terms of this, where he came up with this original idea of plasticity. But James is really important to notice that, of all our body systems and so on, the brain is the most changeable kind of one, because that's how we learn, that's how we can plan things. That's really a fundamental property of what the brain does. And we're really just starting to understand how to exploit this and understand how plasticity might be a problem in schizophrenia. So I showed you these packages of messengers before. Could be dopamine, could be other messengers. GABA and glutamate are important messengers. What we're really interested in, though, is studying these kind of proteins that are involved in how the messenger gets released and can talk to the next cell. So what happens is these three key proteins have to come together, and they have to form what we call a protein complex. They all interdigitate together like my fingers are going together. That brings the package close to the membrane, and then we actually have the membranes join, and the package can release its content. So part of what we think is involved with plasticity is this whole mechanism of how the cells communicate. Not just the messenger itself, but the mechanism of how that information is released, how sensitive it is. Is it too sensitive? Is it overexcitable? Or is it not, should it be inhibited? Should it be turned down in terms of how it works? So this is one of my technicians. We actually look in my lab at these proteins in postmortem brain samples from people who some had schizophrenia, some had Alzheimer's, some had no neuropsychiatric illness. So we can understand about these proteins. There's a little video clip coming up that shows us dissecting out a piece of the brain. The area that we're looking at is called the hippocampus. The hippocampus, because the old Greek anatomists thought it was shaped like a seahorse. And so what I do, get these samples from people in life who consented to donate the samples after the death, obviously. And so we dissect out well, you'd be surprised, but sometimes, no, we have to wait. Anyway, so we dissect this out. It's on dry ice. That's why there's the kind of, smoke coming off it, because we have to keep everything very cold. These proteins are quite stable, provided you keep it cold. We take out that little piece of the hippocampus, and then what we do afterwards is kind of grind it up, make it into a solution so we can measure those proteins in the piece of tissue that were there in the piece of tissue. And what we know about these proteins is they form these coils. They really come together, just like my fingers would be coming together. And remember, that allows the messages to go across. Now, if your system is set up well to allow these messages to go across, it'll obviously work a lot better, and it may need to change in order that a memory can stick with you, that a memory could be retrieved this is part of how plasticity works. And what we can do, this is my postdoc Alfredo Ramos Miguel's work, he's developed ways that we can measure these proteins, but not just the individual proteins, we can actually measure them coming together, which is pretty exciting actually for us in the lab, that we can get at some of the function of what these proteins do by measuring them. These colors show where the proteins that are coming together are and what the composition of these complexes of proteins is. So we can do that to give us a clue about plasticity in the people in the brains at that time. You know, that they had their plasticity prior to death, really. So this is looking, and we have a, you know, this kind of science. We test a hypothesis. We see, could we be wrong because of this reason? Could we be wrong because of that reason? We try lots of things to prove ourselves wrong, actually, before we're really confident that something is meaningful. So what these graphs are showing are two cohorts. A cohort's a group of samples from people who had schizophrenia, people who didn't. So this is from cohort number 1. This is from cohort number 2, because maybe there was a bias where the samples came from or something. And we also then, in cohort 1, we looked in 2 different places in the brain. This is samples from more the frontal lobe of the brain. This is more from the sort of the center, called the cingulate gyrus. And what we're doing is we're measuring all the individual pieces of the complex with our tags or our markers and seeing when they come together. And so what you can, I'll just highlight with this, we measure this component, this marker called syntaxin. We measure that one, and we see that in the schizophrenia samples pretty consistently. The overall group had more of this marker, which is telling us they had more of the whole complex. And then we do the study in a different way. We measure this part. We measure this protein that comes together. This is our tag. And again, you can see, same thing. You know, we're trying to prove ourselves wrong, but no, it holds up. If we measure by this one, we get the same effect of the schizophrenia tissues had more in it. And same if we do the third one of these partners, these 3 partners that come together. Again, it's the schizophrenia samples that have more of that thing, which is telling us that the overall partnership is too strong. There's more attraction between these proteins that are involved in plasticity. That attraction, for some reason, is higher in the brain tissue of people who had schizophrenia. And this is contributing, we think, to the abnormal plasticity that they have. So if we kind of put a magnifying glass and blow up that part where remember this has got a dock and release its contents, if we kind of blow that up and look at those proteins, what's happening is there's too much of these complex coming together. And what we'd really like to do is block that somehow. Okay, we'd like to knock that down, decrease that amount. Now, there is actually, just to think of it, there's one way to do this, which is used in private clinics and actually was discovered in Vancouver. There's one way to do that. It's called Botox. That's how Botox works. It chops these proteins apart. And that's why you put that in your forehead. It relaxes the muscles permanently, for a number of months, because the message can't go across. You don't get those lines because the message can't go across from the nerve cell to the muscle cell because the Botox has cut those proteins apart. Now, that is unfortunate. Well, actually, we do use that in neurology to some extent, even in patients with schizophrenia who have bad side effects with some you may have seen with medications where they have a muscle that just won't relax. Sometimes, not often, but sometimes the neurologist will use that. But we can't use that in the brain. I mean, it's like it's botulism, right? Botulism can kill you. The reason botulism kills you is because your muscles of your lung stop working, so you can't breathe. That's the same sort of mechanism attacking these proteins. Now, what we would want is a kinder, gentler way to do that, if you will, with not with a toxin like botulinum toxin, but if we could make a medicine that would just decrease a bit, and just in those places in the brain where there was too much interaction. That would really be ideal. So we are actually in the lab working on this now. So those are our three, remember, those are our three partner proteins that come together so the vesicle can talk to the next cell. If you imagine, I do this in in a dish. These are my capturing molecules that'll pull these out of the brain tissue. These are my detectors. So I've got my capturing ones. Now, just imagine if they were trying to come together, but we had that red drug that would block them coming together, not cut them to pieces like the botulinum toxin does, but just block them coming together. Then my test would give me less signal because it would be saying that this molecule that you tried is effective in decreasing that interaction. So this is how we discover drugs. This is how we characterize new molecules that would have the actions that we're interested in. Now, usually I don't do this entirely on my own. Go to this place, which is the new Pharmaceutical Sciences building on the UBC campus. It's a really cool kind of building, I think. In there now they have the Center for Drug Research and Development. So we worked with the CDRD group. And in this first study, we screened 26,000 molecules. They just come on plates, and here's a library. It is a library of molecules. We screened ones using that same kind of test I showed you of catching the molecules and trying to see if one of them is decreasing the interactions. And we found some, these just rare ones here, that decrease the amount of that interaction. So those are potential molecules. They're not drugs yet. The potential molecules that could be helpful to decrease this amount of interaction. So the next step of that is saying, well, is there a dose response? Because any medicine should have a proper dose for doing what you think it's going to do. And so what we'd like to see, and some of the molecules we looked at did show this, we increase the concentration and there's more effect. Some of them didn't. It's kind of, we throw those ones away because they're not so interesting to us. It's the ones that show kind of a dose response that get us more excited. So we did that, and this is just, again, focusing now on one of the molecules. This is those, what you see here, this shows the amount of those proteins interacting. So high is lots of the proteins interacting. You can see that in the schizophrenia brain tissue, just like we showed before. There's more interaction than in the brain tissue from people who didn't have schizophrenia. And then we add a little bit of our new molecule that we're working with, 10 or 100 micromolar of that molecule. And you can see that makes the proteins in the normal brain less interaction over time. And it has the same effect. The schizophrenia proteins are kind of more resistant to it, but they still do comply. They have less interaction over time. You can see that by adding 10 units of this drug to the schizophrenia brain tissue, we bring it down to what the normal tissue was. So that's, again, these are the steps we take that are really quite encouraging for us, that we might be on the path to have new medicines that would work in a totally different way in the brain to change the plasticity at a molecular level of these proteins interacting together. We've done a second screen. We looked in another library. It had 100,000 drugs in. So we're still at the stage of pulling out the ones and refining our candidates through this kind of work and other experiments to try and get down to the ones that might be novel and interesting. Another thing we're going to do, though, probably this summer and late fall, is look at known drugs, because there could be some out there that maybe are used for cancer or for something else. Maybe there's some kind of antibiotic or something that might have this effect, but we've just never schizophrenia or for a neurological illness. So if we can identify in a dish that a known drug has these same kind of effects, then that would be, we're able to jump over all the problems of having to do animal testing and toxicity, and because these known drugs are safe drugs. So maybe there's something out there already that we could try, which would be a really wonderful shortcut if we can find something. So that's kind of the next year's kind of work. And if we pull something out, then we could do clinical trials fairly quickly This is a long haul. I mean, this is 5 to 10 years to get a molecule that nobody's ever made into a drug yet up to being able to be used in humans. That's a long ways to go. But, you know, I'm firmly committed to it because I think it's important enough, and I want to do this kind of work. So that's the first bit of the talk about the kind of the brain science of plasticity and so on. Now I want to get to this issue about shrinkage. And that's our friend the hippocampus here, the sort of shape of Greeks thought it was like a seahorse or so. And the hippocampus is very involved with learning and memory. It's actually one of the first things that goes in Alzheimer's disease. But it's important in all of us, but in schizophrenia too. And this again just showing this is a post-mortem brain where the hippocampus sits. These are the blood vessels that supply it. And we can see it actually on MRI scans. My colleague Alex Rauscher and Donna Lang did this scan at UBC, which we can really now with the MRI techniques even start to see the blood vessels that are supplying it. So with another colleague, Ken Sawada, we looked at, these are just the brain scans across here, and the colors are the parts of this hippocampus that's involved in learning and memory, and we can measure those things. And what we tend to find, literature would say, is that they're a bit smaller in the brain in schizophrenia. And what Ken did was then measure these individual parts of the hippocampus. So this is healthy people. This is first episode schizophrenia, so young people at their first episode. And then this is chronic schizophrenia. This is the volume or the amount of the hippocampus brain tissue. You can see there's a lot, big range, right? Mine might be smaller than yours, whatever kind of thing. There's a big range. And so it's not an imaging test that we can say, oh, you've got schizophrenia or something. Can't do that for individual people, but we can see that on the whole, healthy people have a larger hippocampus than people even at the first episode of schizophrenia. And it seems with the more chronic patients who have obviously had the illness for longer, that there is further maybe shrinkage. We can't tell from this sort of thing whether they always had a small hippocampus to begin with, but when we see that the first episode group was larger than the chronic, we think, well, maybe it did narrow down and shrink a bit over time. We looked then at this in different ways to try to figure it out, and we found that, yeah, the longer you've had the illness, there was a relationship between the size of the hippocampus and the duration of the illness, not just looking at first episode or chronic, looking at the amount of time. The other thing that's really interesting from this is that schizophrenia has kind of, we think, two broad kinds of symptoms, right? There's the positive symptoms, the hearing voices, having delusions, those kind of things, but very disabling for patients and very challenging for families too, are what we call the negative symptoms. So that's the loss of drive, the kind of blank, loss of emotional expression and so on. Those are the negative symptoms, and they're really tough. So what we found is that patients, if they had more of these negative symptoms, if it was more severe, parts of their hippocampus was actually somewhat smaller. So there was a relationship, an association between smaller size and more of these negative kind of symptoms. So that tells us, when we find a connection between a structure and a symptom or a function, maybe it really is important. The final thing then was, well, what happens over time? So healthy people, from the first scan to 6 months later, there's no meaningful difference in the size of their hippocampus. But the first episode patients, even over 6 months, they went down a little bit. So that's telling us that there probably is something about the illness process or about their experiences in life over those 6 months that wasn't a good sort of thing, and their hippocampus volume went down a bit. Don't get too worried, because there's a happy ending to this story. Because that's what we want to know. Can we reverse some of this brain shrinkage? I mean, most of the literature in schizophrenia and some of my colleagues emphasize the progressive aspects and emphasize the deterioration and those sort of things. There are. Clinically, we all know that. These are not small problems. They're real. But there is reason to be more optimistic. Can we reverse some of this shrinkage? So this is a ways back now. That's actually me at Whistler with my friend Peter Falkei. He's the chairman of Psychiatry in Munich now. So we used to exercise our first couple of conferences, but then we got a little older and we just kind of have coffee and actually, no joke, this was 1998. We went on a walk after coffee and said, is there a study we could do together? Something we could look at, particularly around the brain. And you know, at that time, and some of you may remember I showed some of this a little last time I was here actually, but I'm giving you an update too. At that time, we were really excited from the basic science that was saying, you know, the brain cells, they can still divide. You know, when I was in medical school, we were taught, oh, the brain cells, once you've got them, you just lose them. Or you can maybe keep them, but they don't grow anymore. Well, you know, that was changing. They were showing in animals that the hippocampus could actually have new brain cells forming, and more importantly, I think, new connections. Because plasticity, we're all about connections, right? We're not just about the number of cells, we're about their connections, what those are doing. So they found that by having animals exercise, they could actually get see more of these nerve cells and more of these connections. So Peter and I were thinking, well, gee, could we get some patients with schizophrenia to exercise? And with these MRI scans now that, you know, we can measure the hippocampus and those sort of things, could we see if there's a change or not? And we thought it might be impaired, you know, that we thought maybe healthy people will increase their hippocampus, but schizophrenia, I don't know, you know, but let's try. So that's what we did. Peter's group did. I helped them out with some of the measurements and some of those kind of things, but this was his group work done in Germany. So the idea was then we got healthy people who were kind of middle-aged, sedentary men, and we had them have an MRI scan and then exercise 3 times a week for 3 months on a stationary bicycle. We thought schizophrenia patients, we can get them to do that too, but really important in any studies of schizophrenia, you want to make sure you're not having a non-specific effect. Because, you know, some of the lifestyle of patients, it's like you do anything, they're glad to see somebody come by, they're glad to get involved, and maybe it's a non-specific kind of thing. So we thought, yeah, we'll do the exercise part, but we'll also randomize, and they sort of, you know, flip a coin, if you will. Not exactly how we did it, but, and maybe they get another intervention, which is something different for them. We thought, you know, being Germans, they would do table football. They come together in a group. They have a trainer who's, you know, would sort of accompany them for the table football and those sort of things too. Because that's an intervention that we thought would have the non-specific effects, but not the aerobic exercise piece, and do this kind of comparison. Whoops. So that's again just how we measure the hippocampus on the MRI scans. Peter's group did that. And here's what we found. So these are healthy people, sedentary guys, 8, before their hippocampus size, after the exercise. It goes up. It went up by 16% actually, which was really, I mean, we were surprised to see this amount of increase in size. Now, you know, the brain's inside your skull, so the brain can't get too much bigger, your head blows up, right? So it's not, you know, there was enough room there that it could get that much increase, but that's a really, you know, pretty impressive thing. Now look at this. This was really just, we were, you know, astounded. I mean, this is before, so the hippocampus tended, even this only 8 men, but tended to be a little bit smaller, which was not unexpected, but it went up in size. It went up by 12%, really quite substantial, a change in that brain structure. Not in every patient, but you know, it was really statistically quite encouraging. And in the group that had the table football, they enjoyed it, they went 3 times a week, all that kind of stuff. Their hippocampus didn't change in size, okay? So we really think it was the aerobic exercise part, not the doing something in a group or having, you know, the Fraulein instructor or whatever kind of with them. Well, they're young guys, you know. So it really was a finding that we're very excited about. Now, it's not, this is another, these are images of brains showing the colored spots are changes where the brain got a little bit thicker as well. So it wasn't just in the hippocampus, there were other areas too that had some expansion of the thickness of the brain, which we think in this case is a good thing. So that was really quite exciting. Now Peter's group, myself included, have done another study. That was exercise 1. This is exercise 2, which just was published this year, looking at particularly the cognitive symptoms, because we know that patients with schizophrenia, their cognition's a little bit down, and these negative symptoms. We already talked about those negative symptoms earlier. And the negative symptoms, this is the endurance training, again, compared with table soccer. The negative symptoms came down, not at 6 weeks, but after 3 months of being involved with the endurance training. There was I think a meaningful change in the negative symptoms, improving those kind of symptoms. We think that exercise is good for depression. Negative symptoms kind of overlap sometimes with depression, but really we think they're a distinct kind of thing, and they are sensitive to this improvement. They improve and are sensitive then to the aerobic benefits of endurance training. So the next group I collaborate with, anybody know what this city is? No, not New York. It's this building here is pretty distinctive. This is Hong Kong. So I collaborate with Professor Eric Chen. He's the head of psychiatry in Hong Kong. Eric runs a first episode psychosis program, covers the entire 7 million people of Hong Kong. They're spread out in 4 or 5 different sites, hospital-based, but in the community as well. It's really a wonderful program. Eric was very interested in the study that we did. I'm sorry, I didn't quite finish this one. Wanted to extend that because our study was in, the German study was in men who were kind of chronic patients. Eric wanted to look in his first episode sample, wanted to focus on women, because women are sometimes underrepresented in our research, and look at women with stable symptoms who were on medication treatment. And did 2 different kind of interventions, actually, because he had a yoga master who was one of his graduate students. So they actually said, well, let's try yoga. I mean, is exercise, just aerobic exercise, good for the patients, or might yoga do something? But we did have an aerobic exercise group. And the third group, because this is randomized and this is research, we said, well, you have to wait. You have to wait 3 months, and then you can go either in yoga, we'll give you the yoga program or the exercise program, but you've got to wait. Because we had to have a comparison group who didn't get any kind of immediate intervention. Now, Eric was initially very interested in cognition, in thinking, in learning, and in memory. Because it's not down in schizophrenia like dementia or Alzheimer's disease or so on, but it's down a little bit. And what they found was that both after 3 months of either yoga or of aerobic exercise, the memory scores improved in the patients. They went up a little bit, but not meaningful in the ones who are on the weight group. Part of the reason they go up a little bit is just you practice, right? First time you have a memory test, you don't do so well. You come back in 3 months, you've done this test before, you're not so nervous, your score goes up a little bit. But that's the comparison group. That's why it's important to have a comparison group. But you see how much the yoga and the aerobic group went up a lot more. And it stayed, which is really exciting, because a lot of the interventions we have, some of the psychological interventions and so on, we worry that they won't persist, they won't last as long. But it seemed in the yoga and the aerobic group that those interventions did persist over a year and a half. And actually, I didn't put it on the graph, but the waitlist group, after they had the intervention, they go up to about this level too. But that's not so it's good for them because everybody got some benefit out of being in research. But the important part is this change over the 3 months, and the fact that persists for some of the memory deficits that the patients had. Now this is showing, back to our negative symptoms again. We had some evidence about these negative symptoms that they were related to that hippocampus. In Dr. Sawada's study, the shrinkage of the hippocampus which we could reverse partially with exercise. The shrinkage was related to more severe negative symptoms. Well, exercise, either the yoga or the aerobic exercise, again, in this study, like in exercise 2 in Germany, was showing a decrease in the negative symptoms, which again persisted over time out to 18 months. So this is really quite encouraging for us that some of these things do indeed seem to be reversible and not damage, if you will. They're plastic. They can change over time. The hippocampus did increase very, very slightly. And this is work, my lab did the measurements and so on of this. It's statistically significant. The weightless group went down a little bit. The aerobic exercise group went up a little bit. The yoga group didn't change, actually. So we were happy about that, but there may be other effects on the plasticity that are not just dependent on this brain structure. So whatever reason, we're glad it works and so on. That was statistically significant. Now, what they've done in Hong Kong, which is just really, it's an incredible city to visit if you ever have a chance or so, but what Eric's team has done throughout their whole program, they've introduced fitness and either yoga or aerobic fitness, and give you that website. It is in Cantonese, but there's lots of videos and things you can look at if you're interested. They've done these real, group kind of things. They've brought in even family members and so on to help encourage with the patients and really participate in. They've developed routines of kind of movement, almost like dance in some ways, and they work this out real carefully, though, that if you do these things this many times, it gets your heart rate up this amount, because for exercise you should increase your heart rate by this amount. So they've got this all figured out really well, and really are implementing it throughout the special administrative region in terms of this as part now and parcel of their early psychosis programs, which I think is really exciting and makes Peter, myself, and Eric feel really good about having something come from kind of academic research sort of studies that can be implemented and hopefully will be improving the outcomes that we have in people with illness. So I think this is, showing us that at baseline, and those of you who have family members and so on probably know all too well, the aerobic fitness is often a bit low. That this schizophrenia does have these kind of smaller hippocampi, but with support, and they're finding that patients can be encouraged to be in exercise, and at least this part of plasticity in schizophrenia, schizophrenia maybe have some impairments of plasticity, this part is amenable to an intervention. So if we get the interventions right, maybe we can overcome some of these problems of plasticity through medicines maybe, but also through the everyday life kinds of things. And really we're optimistic that the cognitive function and perhaps some of those negative symptoms could be improved through these kinds of interventions. Promising add-on treatment for patients who are on medications as well, taps into that reserve of plasticity that is intact in the illness. And may not just be aerobic, it may be, we can tailor exercise, or as one of my colleagues in UBC likes to say, prescribe exercise to patients, and a specific prescription that fits well, I think is really important to go forward. Okay, I'll try and go through. It is warm in here, and people want to leave some time for questions. Just give you a taste of a kind of really, for me, interesting idea. Everyday life kind of thing. So where's this? The bullet trains. This is Japan, right? Yep. That's where you can get great ramen. And that's my friend, Dr. Ken Sawada and his kids. We're visiting them. Ken's a very creative guy. So in his wards, he was, it's Japan, right? So the patients even do karaoke. And Ken was like, the sound of the karaoke that the patients do, it is so awful. I don't understand this kind of thing. So I said, well, maybe their musical ability, maybe schizophrenia actually impairs the musical ability. And so he did a study. First, he looked at their cognitive functioning. He looked at cognitive functioning in healthy people. Then, as we expect, the cognitive function's a little bit down in patients with schizophrenia. And then he said, well, is there something we can do to test their music ability. And actually there is. This is a test that was developed in Montreal, actually. The Montreal, McGill University Psychology Department, they are like the world leaders in understanding your brain on music. They do wonderful work with this, and they develop some tests. And what the tests work, and I'll hopefully be able to give you a little taste of this. They play some sounds, some music, and then they play some after it, and you're asked, was it what you heard the first time the same or different from what you heard the second time? And I understand music, but they're designed to test all these different aspects of music. So hopefully we'll get a sense of this. Same or different? That's what you're asked, okay? It seemed different to me, too. All right. Some saying different, some saying the same, some sort of. I'm not sure the actual answer. Those must be. Do one more. Oh, okay. Well, so that is, give you a sense, right? And so the test goes through. It takes 15, 20 minutes to do, but all these different kind of things, which is tapping into our musical ability. And one of the things I think that's really exciting about music actually is that it's a unique human thing. I mean, we'll never really have animals that have schizophrenia. They can't talk, they just don't have the same experiences as we do as humans. And schizophrenia in some ways is a very human specific kind of illness. Just like language is a specific human kind of ability, music may be as well. So it's just really neat to think about. Now, so this is how women and men fare on this Montreal test. Women, obviously, they do a bit better. They're better at lots of things than men. And that's meaningful. Same thing, schizophrenia though, we got a drop in terms of their score, Ken did, in his patients in Japan, on this test of musical ability. Still there was the better performance in women with the illness, but it was down, their ability to perform on that music kind of test. The global score in the music and then these different aspects of music and so on, they were all decreased in ability of the patients with schizophrenia. And we find in healthy people, the dashed line, there's some decrease with age in musical ability on this test. The decrease was steeper, actually, in the patients who had schizophrenia. So something really is going on with this that I think is really quite interesting. And there was an association between the score on the music test and the scores on the cognitive test. So to me, it's almost an alternate way of assessing somebody's cognitive function is how well can they perform on this music test, which I think is really pretty neat. And look at this. The score on the music test is high when you have few negative symptoms. More negative symptoms that you have, the score on the music test is lower. So it's tapping in maybe to some of those areas of plasticity or of impaired function in the illness. And we found that the music ability has partly contributions from the cognitive functioning and partly independent contributions from these negative symptoms that patients have. Now, okay, there's abnormalities in music ability and so on, but what about music therapy? Because there are music therapists. In fact, on the ward that I consult to for refractory psychosis out in Detweiler at UBC campus, we have a music therapist. And I admit, I was initially skeptical, but we have her for a half a day a week, and she's actually outstanding. So this actually is showing us, you don't know all the details of this. This is a study that was reported of patients who had a stroke and whether or not they had music therapy after their stroke. And the patients who had the music therapy actually improved a number of their cognitive functions, and these were damaged, obviously, by the stroke. So if you look in the literature, and this is the Cochrane database, the Cochrane group in the UK, they're the most rigorous people of all in terms of saying whether an intervention, a treatment makes a difference. Almost all the time they end up saying, oh, there's maybe a little bit of difference, but we need more research or whatnot. This one, there's not a lot of data, there's not a lot of studies or whatnot, but it shows that music therapy, and the way you read these kind of things is you look at this bar here, and the way you look at it is anything to your left, all these study dots on the left, if they're on the left side of this line, that means they worked. And actually, there's only, not a lot of, this is comparing 129 people in all the studies in the literature who had the music therapy compared to 111 who didn't. So this is small. I mean, drug trials, we have thousands of patients and whatnot, but this is small. But nonetheless, to me, it's actually quite encouraging that maybe this kind of intervention, and we're talking about everyday life interventions, it was helpful, and particularly this analysis of the data was for those negative symptoms of the illness. The Ken study shows is related to their music abilities and so on, that our work on exercise shows do improve with that kind of intervention around the plasticity. So this is, again, just very early, very preliminary. My colleague Dr. Alistair Barr is working on trying to do some of this music-related work at UBC on our ward. These are some of the most difficult patients in the province, so we'll have a chance to see just how much of a difference it may make. But to conclude our talk then, these old ideas I started about the dopamine, that's important in the everyday lives of patients. There's no question. That's how our medications work. They prevent relapses that way too. But we need, I'm very keen on looking at these new ideas about the ability of the brain to change, not just to block the neurotransmitters and so on, or stop using the stimulant drugs, but what can we do? Can we make medicines that help speed up some of these changes? And what changes in lifestyle, maybe even in terms of exposure to music and those kind of therapies, could make a real difference for our patients in new ways of going forward. So I had a lot of contributors and colleagues in this work at UBC, Simon Fraser in Germany, Peter Fakai's group from Munich and Göttingen, Ken Sawada's group in Kochi in Japan, and Eric's group, of course, in Hong Kong is really critical to these sort of things. So I thank you for your attention. I welcome any questions. Yeah.
I was wondering on your study with the yoga and the aerobic and your follow-up group, was there any way to sort of monitor whether they continue to do that on their own, or I guess you stopped them giving the program?
Yeah, and the details from Eric, I'd have to check back on that. I think some did still participate in the programs and so on. With the aerobic exercise, in terms of the brain structure, we do see that it will slide back towards what it was, but some of the benefits, could be longer term without having the brain structure change, because it happens elsewhere in the brain and at more subtle aspects of plasticity than whether or not your hippocampus changes. But largely, I do think you have to keep up with these lifestyle interventions and whatnot, to maintain the gains that you've made. But some of the things we don't know are to what extent for example, with aerobic exercise, is it the increased muscle power and the increased fitness of oxygen, consumption, all that stuff, or is it just the fact that you're moving? And you're getting, because movement causes more muscle inputs to the brain that stimulates the brain in a way too. We haven't yet begun to dissect out what is important about the exercise interventions, how we might match different kinds of exercise interventions to different kind of people. I mean, Eric chose aerobic, in Hong Kong, the initial idea was just to do yoga, actually, because young women in Hong Kong, they like yoga, they're willing to do that. Everybody knows that for ourselves and for our family members, it's hard to do exercise, it's hard to do that. So they thought, well, let's try yoga, and I convince them to do the aerobic as a comparison group or so on too. But we don't know yet, and we don't know just to what level, whether the maintenance amount of the intervention is the same as that initial amount or so on. Just don't know that yet, but they're really important things to find out about.
The actual program stopped and they
So the experimental program, yes, that 3 weeks of 3 months of structured thing, yes, that stopped. They didn't continue. We didn't have the resources to offer that continually for that time with the monitoring and all that kind of stuff that was involved with the active one. That's right. Yeah.
they continued and then they
On their own, yes, yeah, it would be Stacy.
Just wondering, the kids that do a lot of marijuana, I'm assuming that if the adolescents that have potentially hippocampal changes and then become schizophrenic, do you think that's a link to structural change or not?
So there's a lot of concern about cannabis, and particularly in the UK, Professor Sir Robin Murray has been very adamant about the risk that is related to high potency cannabis in the UK, feels that it's almost causative for schizophrenia. In British Columbia, probably 60-70% of our high school students are exposed to cannabis at some time. We do not have an epidemic of schizophrenia in the province. However, I'm more and more convinced that there is a role of cannabinoids, some cannabinoids, in increasing psychosis. I mean, my other major studies in the downtown east side, where, as you know, people are using heroin, using methamphetamine, using crack. They get psychotic from some of those stimulant drugs, but actually the ones who use cannabis are just a little bit more psychotic, actually. We can even see it in that population, that it creates an edge. So I am concerned about it. Cannabis is a complex thing, though. It's a plant. There are 90, 100 or more active ingredients that are being ingested when someone smokes or consumes marijuana, cannabis, or so on. There are different types. There's indica, there's sativa, there's all sorts of things we don't know about it. We don't know what, some ideas in terms of the THC content probably is what is the activating and psychosis triggering kind of part. There's other parts, though, the cannabidiol, that there's some evidence actually that it can be helpful. Different strains of different amounts. You don't know what you're taking. You don't know what it is really. I mean, now that it's become medicalized to some extent, we hope, and it's not medicalized like medicines are, because we still don't know what the active ones are or whatnot. It was interesting. I was at a conference once where there were some Dutch psychiatrists, and they've had the cannabis cafes for a long time. And so they would say, well, these patients, they're on clozapine, they're really, most severely ill, they still want to use the cannabis. We tell them, well, at least use the brown stuff, not the yellow stuff, because the brown stuff won't harm you as much. So there's a range of these sort of things. Now, how does it do that, though? Is it through, we don't think it's that dopamine rush or push out like the amphetamines and the stimulants do. Is it changing some of these plasticity mechanisms or so? Maybe. It's not clear. I don't know that the evidence is that good that brain structure itself changes with the cannabis. I think of it more in a chemical kind of way or so. But it's really, we're too early days, and I do hope to some extent that it becomes a little bit more medicalized, if you will, because I'm very interested in trying to dissect out some of these things, to isolate what parts of the plant are dangerous and are leading to psychosis, what parts of us, because we're all different, we have different gene variants and so on. Maybe some of us are more sensitive than others. We certainly hear, some kids who I took that once and I'm never taking that again because I felt really bad. And other kids will, go into using it regularly. So there's different variation within us. There's different variation in terms of what people are ingesting. It's totally unclear. It's not a good idea for people who have the illness to use it. But
Brain, like if you got an 18-year-old doing really good exercise versus a 40-year-old, do you think there's a chance that they could have better neuroplasticity because their brains are younger, or don't? Sure, you don't.
Hard to say. You know, I mean, in the first study, the exercise one in Germany, those were 40, 45-year-old men with chronic schizophrenia who we saw the biggest increase in the plasticity in that measure of their brain structure. Now, the younger women in Hong Kong, they didn't have so much of a change in their hippocampus, but it was probably pretty plump, you know, pretty healthy. You know, they were younger, those sort of things. So some aspects of plasticity, yes, maybe better in terms of the cognitive uptake or the decrease in negative symptoms. Maybe those are better in younger people, but other aspects of plasticity are still there in all of us. You know, it's that connection in of our frontal lobes that's involved in planning and, you know, reasoning and so on. That really isn't happening till our 30s or so with the final levels of those connections to the other parts of the brain and whatnot. So these things are still maturing. You know, I think of development goes up to age 2 or 3, and then we mature after that. And some things, again, it's another talk and that kind of stuff about aging and dementia and so on. I think this idea is that we all lose cognitive function as we get older. No, we don't. Cognition changes. Some skills that we had when we were younger, we may not have when we were older, but there's other kinds of ways that experience is integrated in older people that younger people can't do as well. So, yeah, you can make you know, broad statements about that. Aside from, there's probably aspects of plasticity that are easier to tap into in younger people, and there may be aspects that are better to tap into older people. I'm glass more than half full on this kind of thing, for sure. Yeah.
I was curious, there's been a lot of studies about meditation and brain plasticity, and I was wondering if there's ever been studies about that in relation to schizophrenia. The other question was, could you elaborate on music therapy, what that entails?
So meditation and schizophrenia first. There is actually a meditative component to many yoga, as I understand, to many yoga type treatments. That was not included in the yoga exercises that the patients did in Hong Kong, because there's some, I think, thought that for patients who do have schizophrenia, meditative experiences may not be the best thing for them. Now, I don't know anything about this kind of thing. That's just why the graduate student, who actually is a qualified yoga practitioner and teacher and so on, she decided that for schizophrenia we shouldn't use that meditative component. For other mental disorders and so on, anxiety disorders or depression, maybe the meditative component is more important than the flexibility components. I don't know. I'm really not sure about that, but I do know we didn't use that for that reason in the schizophrenia study or so on. Music therapy, I must confess I don't know a great deal about. I mean, our music therapist, I'm just struck though, our music therapist on the ward, and again, we go through did this morning, our multidisciplinary team round. We find out, about the history, the diagnosis, how the patient's doing in the ward, and these are some of the more severely ill patients in the province, and we do our best, Dr. Menon, to engage them in cognitive behavioral therapy and those kind of things to complement the medicines we're changing around. But often enough it's, well, they just can't concentrate enough, they can't do the CBT. But Moira manages to get them into the music therapy groups and so on. So, I mean, I don't honestly know, but the things that she does assess are their ability to follow rhythm, to do these kind of things, and then they do have, group sessions on just experiencing music and doing those kind of things. Music is really interesting too, because there are these sort of procedural things, if you will, in terms of the rhythm, the beats, all those scales and things, but there's an emotional component to music too, and how it makes us feel. There's songs that can make you feel very sad, there's songs that can make you, feel energized or spiritually better and so on. So there's a lot of things I think one can do with music, but I'm totally naive about this. It just seems to me, from the basic understanding that this is an impairment in schizophrenia, maybe it's an opportunity to try and do something that would be therapeutic. And, Sarah McLaughlin's group or something wants to help us out or whatever, we'd be happy to talk to them, I'm sure. There and there. So first.
I was wondering if there's been any significant cross-cultural studies of, like, this
So question was about cross-cultural studies and does I guess the prevalence, how many people in a culture or so on have the illness, is that different? We don't think there's that much difference between cultures or ethnic groups in terms of how much there really is there. The amount of stigma around the illness does differ amongst cultures. And we know that stigma is a problem that leads to patients and their families maybe not seeking out treatment or having barriers to treatment and so on. And certainly, from my experience working with Eric in Hong Kong and in China and so on, in Asian cultures, there still is a lot of stigma attached to schizophrenia. So that barrier differs more than the actual illness does, I think, itself. I mean, we have First Nations, patients who, again, have a totally different background and experience, and it is there in their communities and so on, too. I think some of the things we're learning about the illness are more the experiences people have in dense urban environments as opposed to rural environments. So countries where there's less density and those kind of things, less, if there are those countries anymore, but because so many people are moving into cities and into high density environments and so on, that we do think do increase the risk to some extent. Immigration, those kind of things do increase the risk. We did, there was a lot of concern about Afro-Caribbean immigrants to London. They had a higher amount of schizophrenia. We look back with our Riverview records at people from England immigrating to British Columbia in the late 1800s, and they had an increased risk of schizophrenia too. So it wasn't the ethnicity being Afro-Caribbean or being English, it was more the action of going to a different place. And the stress and I think, our sense of it now is the social stress that happens, because, again, you read this stuff in the late 1800s, early 1900s, British immigrants to British Columbia were not treated that nicely by Canadians who were here before. There were newcomers, that same sort of thing as immigrants often experience wherever they've come from. So it is that specific, I think, kinds of stressors that are... I don't know. Or the Irish. We've got our new Dean of Medicine's coming from Ireland, so we'll have to, well, he's Irish, he's coming with the UK accent. Watch, I didn't say that. No, he's really looking forward to having our new Dean, actually. So yeah, those are important things, though, that we do understand. Again, that's, I think, a great opportunity for us in the Lower Mainland and in British Columbia to explore some of these things more, because we do have such diversity here, and it's a great opportunity for us to do some of that kind of work. It's a good question. I think you had a question, and then we'll go back.
I'm just curious, you've shown the statistics look very promising. I'm just curious, from the people that you did the experiment on, was there an improvement enough that there was any cognition that I feel better now?
Yeah, I think there's a general sense of, yeah, I mean, certainly the colleagues in Germany have told me that they, Frank went back and talked to some of the men who had been in the study a year later and whatnot, and they still feel it was meaningful for them. Some of those things have carried through for them and so on. Yeah, I think it is, I am positive about it in that kind of a way. I would say yes. Yes?
That was going to be my question, that there was a decrease in negative symptoms, and I'd like you to understand what those negative symptoms were that you tested, and also, was that done by self-report, or how was that measured?
So the negative symptoms we generally look at are apathy, avolition, blunting of emotional expression, so the voice doesn't have as much emotion in it, the facial expressions are down and so on. Those are the kind of negative symptoms that we look at. They're rated by a psychiatrist. They get information from a caregiver or family member or so on to help with that rating, but it's not a self-report of rating or so on. So that's how we do those kind of things. And they're symptoms that are less responsive to medications. Those symptoms are, that we do best with the medications in terms of the positive symptoms. Those negative symptoms are tougher, much tougher to treat and so on. So yeah, I mean, it's a modest degree of optimism, but I do have it for being able to do something about the negative symptoms that often enough, unfortunately, our medicines don't seem to touch that much at all. Yeah. In back, and then back.
Questions about studies occurring or future studies. First of all, have there been divisions of right and left-handed clinical trials studies with people who are either musical or even art clinical trials? Because left-handed people are thought to be more creative to start with. And secondly, has there been any study of the idea of taking normal hippocampal cells, doing a cell transplant into someone with schizophrenia to normalize the protein connecting to it?
Okay, so the first one is a really interesting idea in terms of laterality, left part of the brain, right side of the body, whatever kind of things. Short answer is no, we've not studied enough people, and we generally routinely with neuropsychological tests and so on do measure handedness and look at that. It's a very good idea actually, and I don't know whether we've got sample size, because left-handedness is about 10, 12% of, I'm not sure, even lower. 1% of people are left-handed? Is it that low? I thought it was a little higher. Anyways, whatever. There's mixed laterality. I'm a little bit mixed myself. But anyway, it is something that's very important to look at, and that's just a good reminder for me. So, transplants. So yes, we're, in this era of regenerative medicine, very interested in stem cells, it's kind of cells in our body that continue to divide, replenish our skin, replenish different organs, those few in the brain, those neural stem cells that are still dividing and so on. What we've done so far is some, not we, but medicine has done some work in patients with Parkinson's disease. They lose those dopamine-producing cells. That's what leads to those symptoms, slowing down and whatnot, which are kind of like negative symptoms actually in some ways. But so there have been some studies of transplanting in cells that will make dopamine for, probably there's only been a half a dozen or a dozen patients that have actually had these kind of transplants done. My understanding is that they have been successful in ameliorating some of the symptoms of Parkinson's, but unfortunately there's been some kind of overshoot as well. So there's been more jerking movements and those kind of tremors. So it's not a routine kind of treatment at all yet. Where I think there are optimism around illnesses like multiple sclerosis, where the wrapping of the nerve cells deteriorates, that a transplantation technique might help with that or so on. We're very far away from thinking about whether that would be helpful in schizophrenia or not. That would be considered, I think if we see improvements in neurological illnesses over the next 10 or 20 years, really, or so on, it will take that long before one would even think of that kind of a therapy for patients with schizophrenia. We need to know a lot more about it before going to thinking even about that thing. Which, and two, it's particularly challenging because we don't have an animal model that we can use to understand some of these things before doing something in humans. So there really will be some true breakthroughs before we get to that level of being able to do something, I think. Yeah. There was, fill in the orange and then we'll come here.
Can you touch on the bipolar people and the bipolar 1.4?
My colleague Lakshmi Yatham at UBC is the main person we have in the department on bipolar. I've collaborated with some of his work and so on that. I think neuroplasticity, the things that we're looking at in bipolar are, I think, related to some of the antioxidant mechanisms. things that are, vitamin E and these kind of things are protective for against. There's mechanisms in the brain too that involve proteins getting more of a negative charge on them sometimes, which can be damaging or so. So I think colleague Trevor Young, who was the head of department before me, was very interested in these ideas around bipolar and how we need to understand that, which does seem to be kind of different from schizophrenia, actually. To me, bipolar, it's fascinating in many ways. The patients who are manic can have the same kind of psychotic symptoms as patients with schizophrenia, but once their mania is treated, they go away. Fully they go away, whereas so often with schizophrenia, they're not as pronounced, but they're still there. So there's something different about the psychosis mechanism that's there in both bipolar patients. Of course, the huge difference in bipolar, it's the mood that's the main thing, and the mood goes very high to very low. So there's really quite, to me, still different features in how bipolar expresses itself clinically than schizophrenia does. Some of the risk factors, though, may be the same. We do think that there are developmental abnormalities in both of the illnesses, maybe more in schizophrenia than in bipolar or so, but some of the other stress-related risk factors and these sort of things are similar. Certainly, patients and advocates with bipolar, they do tell me that, exercise is important for them too, and kind of modulating some of these swings and whatnot. So I don't in any way think that plasticity is like some specific thing for schizophrenia or whatnot. It may be beneficial for a whole range of the more serious mental disorders to have those put into place. I think I said up here was next.
I have a stupid question, I guess, but what is the definition of schizophrenia versus bipolar? What's the difference?
So the definition of schizophrenia versus the bipolar and so on, because these was actually, Peter Falkei's predecessor, because Peter being head in Munich, the origins of some of our understanding of mental illnesses were from Kraepelin, who was the chairman in Munich in 1900. So he separated out bipolar being primarily a problem of the mood, okay? And it goes from being very high, manic, elated, grandiose, to going right down through normal to being very depressed, okay? And these are cycles. The characteristic of bipolar is that it cycles. And usually, though, we see very good recovery in between those times of illness, between the cycles of illness. Bipolar people often can do extremely well, business people, those kind of things. It does, okay? So schizophrenia, patients with schizophrenia, they can rarely have manias. The one person we talked about today, I think, had a couple of manic episodes, actually. They can have depression, but that's not the main feature of their illness. Okay, the main feature of schizophrenia is the psychosis, and that's the hallucinations, the sounds, voices that they hear that aren't really there, the delusions, the fixed beliefs that you can't shake that are incorrect, wrong, bizarre kind of delusions or so, and the negative symptoms, the loss of drive, volition, the loss of ability to make contact with others, to have intimate relationships, friendships. That's schizophrenia. Both illnesses have a spectrum of severity, but you're right, the long-term outcome of schizophrenia is not as promising as the long-term outcome for bipolar. But some patients with bipolar can be, not minimizing that at all. They can be very impaired. It can be. So both are really important for us in psychiatry to devote attention to, absolutely. Yeah, those.
Bill, we have
Oh,
someone in the corner.
Oh, yes, okay, sorry.
My question isn't directly related to plasticity, but it is sort of related to diagnosing schizophrenia and the progress, so it's sort of the process of the illness of schizophrenia, and how often, especially in young people, they're diagnosed as schizophrenia and treated for schizophrenia, and then it morphs into a number of things. I mean, from my own experience
And so I'm just wondering about that aspect of diagnosing and medication and the change and the readjustment to diagnosis.
Yeah.
Is that
So,
a fair question?
so, and it comes up to the earlier question too. Part of what the diagnosis of schizophrenia and the psychosis part is, and I omitted to say, is that there should be a decline in function of at least 6 months from a previous level of function, or if it's a younger, really teenager or so on, they don't kind of make some of those gains that we would expect. So Kreplin, the original concept of schizophrenia was this included a loss of function as well as the symptoms. Now, the trouble, I think, maybe in some of these things is that we think that, well, if the function improves, they couldn't have had schizophrenia. And because schizophrenia is always an illness that has bad function and deterioration in function. And that is not true. I mean, yes, at the beginning of the illness, and to make the diagnosis, we have to have seen a decline in function, but that doesn't mean it can't come back. And it doesn't mean that the illness is incurable or it never gets better or so on. So again, not to be totally pessimistic about it. It is, I know, it's my life's work. It is a very serious illness, and most patients with the illness don't have a full recovery back to where they were. But we do see that as patients get into their 40s, their 50s, their 60s, some really do, come back to being not that different from other people in those age groups. The first 5 years of the illness are probably the toughest in terms of that decline of function and just the symptoms and so on are the toughest. And you've got to think too, if somebody develops the illness early in teenage years and doesn't accomplish some of those milestones and so on, well, it's not necessarily the illness directly, it's the secondary consequences of having the illness about missing out on high school graduation that's happening all around us this next month and so on. Those things are really secondary impairments that really have big consequences for people over long periods of time. It doesn't mean they did or didn't have schizophrenia or so on, and I think it shouldn't be, it's just unfortunate because of the stigma and so on. People don't want to say that wasn't schizophrenia or so, kind of thing. It was or it wasn't, but if you're doing well now, great. It doesn't matter so much what the illness kind of was. It is important, though, in terms of, I think, trying to make the diagnosis as well as one can at the beginning or so on. Because, another study we did with Eric's group, we looked at the risk of relapse, because schizophrenia does have, it's not the highs and the lows of the mood like bipolar has, but schizophrenia has episodes of exacerbation of the psychosis, and then they get calmed down again, and we want to prevent those exacerbations. Now, in a person who's got first episode psychosis, it might be schizophrenia, it might be bipolar, it might be something else, but if it's schizophrenia, they're at high risk to have another episode if they go off medication, and we need to keep medication in place for probably more than a couple of years, actually, to allow the brain perhaps to mature more, to accomplish some of those life goals and milestones, which all add up to later on, being less prone to have a relapse. But those aspects of things are important, of getting the diagnosis right, because the non-schizophrenia kind of psychosis aren't quite so prone to have the relapses. Maybe we can get away with less duration of medication treatment for those. So diagnosis, it is important to try and get it right, and it's not easy. It's particularly not easy in adolescence and so on to make those kind of diagnoses for sure. No.
Time for 2 more questions.
Okay, do you want to choose who's? Okay, yeah.
I'm not sure about the name of the alphabet, but I think it's called Graphing Like Moment. I read about something on the website, and they said, I haven't met, I should say up front, I haven't met anyone personally that has tried this for schizophrenia, but I was wondering if you've ever known of anyone who's tried this. Apparently a high level of non-vitamin C, not regulatory niacin, helps with schizophrenia, and I haven't met anyone personally, but I read that it actually helps with schizophrenia.
Yeah, there have been ideas. Dr. Hoffer in Victoria, passed away a few years back, but was using high doses of various vitamin therapies, along with medication, actually. Nothing really proven yet, I think, around niacin or other kinds of vitamins. If anything, folic acid, like take during pregnancy or whatnot, for some patients who have a particular genetic predisposition to not metabolizing folate so well, there's a little bit of evidence in the literature for some patients that folate seem to have some research benefits in terms of their negative symptoms, actually. That's what I'd be most, beside the usual, shower, balanced diet, food groups, all that kind of thing. Maybe there's something with that. Niacin was of some interest 10, 15 years ago, but I've not seen anything really come out about that one specifically yet for most groups. Individuals, can't tell.
There we go, Jim, and then Peter.
Last one.
Mine is a personal related question, which is with concerns to my son. He's 36 years old, and he has had 3 relapses of this illness. And every relapse he was hospitalized. He went on an increased dosage every time. What is the maximum dosage can they go up to? Is there a limit, or they will keep on increasing the dosage?
Yeah. So medicines, when they're approved, have a kind of band. There's a dose you got to get up to have any effect. And then there's a top end that the manufacturer is approved to recommend as the top dose. Physicians are allowed to prescribe, we're licensed, we can prescribe to whatever amount we want. Obviously, if there's a adverse event and you've exceeded what was the suggested guideline, you're taking on bigger liability that your insurance might not cover, kind of thing for that. So, but, again, physicians, we have to do the best we can for individual patients. Going above the maximum of a prescribed, of a recommended dose or something, there are unusual cases that do need higher dose, very much higher doses. Some of us have genetic variations in our livers that chew up the drug, excrete it all out. So that kind of you might need to exceed the maximum doses, but generally we try not to exceed the maximum recommended doses of medicines. Instead, try something else. And for patients who have had a poor response to 2, 3 trials of antipsychotic drugs, we usually would want to try clozapine as a medicine that is the only one that is known to be helpful for patients who have had poor responses to other drugs. So clozapine still is a last of our bag of tricks. If they don't respond to clozapine, it's much tougher then. So those are the things to think about. But you're right, sometimes the higher doses and mixing antipsychotic drugs together is not the best practice. We try to avoid, again, you have to have a dialogue though with your physician or your son's physician to talk about those kind of things and understand. I think it's fair to question them if they are going above to higher doses. Maybe there is a reason that they've got in mind that is specific to your son's case that is worthwhile, but it's fair enough to have a dialogue with them and ask them some of those questions. And certainly if clozapine's not been considered, it's something to think about. Okay.
Bill, this gentleman here was.
Oh, okay. Yeah, one, yes, the encore.
I'm curious about whether the hippocampus was the amygdala or related to the amygdala. I was at a conference with Dr. Lang, Laura Lang, and I think
Yes.
he was working on volumes various.
Yes.
And curious also about a link to intelligence, and also curious, if you will, about whether in all your studies somebody became asymptomatic without medication as an acute.
So the amygdala is another part of the brain. It's a sort of walnut-shaped structure. It just sits in front of the hippocampus, actually. We think the amygdala is involved more with emotional expression, with some anxiety disorders, post-traumatic stress, these kind of things than with schizophrenia per se. We separate that out in our measurements, so the amygdala is measured separately from that kind of thing. In terms of the relationship with intelligence or so on, generally, I mean, I think there's some increased risk of schizophrenia in people who have intellectual deficits, so very, low IQ, intellectual deficit, mental handicap, those sort of things. Their risk of schizophrenia or psychotic states will be somewhat higher. We do think if we measure the IQ of patients who have schizophrenia compared to what we think their IQ was as kids, teenagers, and so on, seeing a gap of 10 points or so is not uncommon. That doesn't classify as being brain damage or anything like that, but it's down a little bit on things. For schizophrenia, if you've got the diagnosis right, to have spontaneous remission without medication would be very unusual. That's not to say that after, in the older years and those kind of things change and so on, but to have a spontaneous remission of an illness that truly is properly diagnosed as schizophrenia would be unusual. Some patients may be able to function in some ways or so on, without medication. That's possible, but the actual illness itself going away spontaneously, not saying it can't happen, but it certainly is unusual, and I've not heard of it. I certainly have met people who are working, have jobs and families and doing just fine, and had 15 years, 20 years of schizophrenia, and they were in hospital for 5 years of that kind of thing. They're on a low dose of medication now, and they're doing fine. I mean, I've seen those. Seeing the kind of case that you described doesn't come to mind, actually.
Thank you so much.
Okay, pleasure.